Hello there, and welcome to the Sleepy Science Channel. Tonight, we are drifting back to one of the oldest mysteries we know of. How did lifeless matter become something that can grow, adapt, and remember? Life's origins cannot be found in a single moment in time. They developed across millennia, influenced by water, the sun, and even other planets. Somewhere between chemistry and biology, the world discovered a way to come alive. Molecules began to team up. Energy got captured, and tiny boundaries formed, allowing careful instructions to be passed on to future generations. What makes this story so captivating is that it is both intimate and cosmic. The atoms in you were once scattered through space. The rules that guide cells today still echo the early Earth, when the sky looked unfamiliar and the planet ran on raw chemistry. If you enjoy these gentle journeys, I invite you to like, subscribe, or share a thought below. It helps others find their way here too, one sleepy soul at a time. But for now, allow your shoulders to soften. Let your breathing slow, and allow your mind to unwind as we wander through the ancient beginnings of our world. Let's begin. The early Earth had unbreathable air, and the sky was a different color. With almost no free oxygen in the air, there was no ozone shield protecting the planet. Ultraviolet light reached the surface far more strongly than it does now, and that harsh light was both a blessing and a curse. It could destroy fragile molecules, but it could also power new chemical reactions that may have fueled early life. The atmosphere was likely dominated by nitrogen and carbon dioxide, with extra gases supplied by volcanoes and meteor impacts. Depending on the mix, the sky would have been hazy rather than the clear blue it is today. It may have even changed colors due to aerosols and gases that scattered sunlight. That haze would have shaped the climate, because it changes how heat is trapped and released. This matters for life's beginnings because chemistry often depends on very precise temperature windows. When you imagine the first steps toward life, try to picture an alien world. One with strange light and unfamiliar air, and a surface full of molecules reacting and competing for energy. Earth's early oceans were likely greenish from dissolved iron in water. Today, the ocean looks blue because light scatters through clear water, and because tiny organisms shape what is dissolved within it. Early oceans were different. With little free oxygen around, iron could stay dissolved and travel widely through seawater. In that setting, the sea could take on a green tint, especially where iron-rich water mixed with sunlight and shallow sediments. That iron was not just a color. It was a powerful chemical player, ready to swap electrons and drive reactions. It could also interact with sulfur and other elements, building minerals that trap clues about ancient conditions. Thinking about a green ocean helps you remember that life began in a world that did not resemble postcards. The chemistry, the light, and even the palette of the planet were unfamiliar. Origins research is partly about learning to imagine that strange, alien Earth. The early sun shone more faintly, yet Earth somehow remained warm. Astronomers and geologists call this the faint young sun problem, and it sits right at the edge of life's earliest chances. With less solar energy arriving at Earth, simple models suggest oceans should have frozen far more extensively than they appear to have done. Yet ancient rocks point to flowing water, weathering, and sediments that imply long-lived liquid seas. The fascination is that the solution cannot be a single knob you turn. It has to be a whole planetary balancing act. The mix of greenhouse gases, the behavior of clouds, and the way oceans move heat around the globe all matter. Even surface reflectivity matters, because darker water and rock absorb more warmth than bright ice. Life's earliest chemistry may have depended on this kind of subtle climate stability, where Earth managed to hold on to warmth despite a weaker sun. Greenhouse gases likely kept early oceans from freezing solid. A thickened atmospheric blanket can trap outgoing heat and keep water liquid even under a fainter sun. Carbon dioxide is the obvious contributor because volcanoes can supply it and weathering can remove it, creating a slow planetary tug-of-war. Methane adds a more dramatic twist because it can be produced by microbes, which raises the possibility of feedback. If early life made methane, warming could increase, which supports more microbial activity and more methane production. Yet, methane can also lead to high-altitude hazes that reflect sunlight and cool the planet, so the story may involve warming and shading in competition. What matters for origins is time. Chemistry needs long stretches of stable liquid water to build complexity. A greenhouse atmosphere, even if it fluctuated, could provide that persistence, turning oceans into reliable laboratories where reactions could run for ages without being frozen out. Life may have begun more than once, with only one lineage surviving. When conditions are right for complex chemistry, it may not happen just once. Early Earth could have hosted multiple experiments in becoming alive, running in parallel across different environments. Some experiments might have produced short-lived proto-lineages that never gained stable inheritance. Others might have been snuffed out by impacts, climate shifts, or competition. The surviving lineage would not necessarily be the first. It would be the one that became robust, adaptable, and able to spread widely. This idea changes how you feel about the common ancestor. Instead of being a singular miracle, it could be the last winner of a crowded early field. It also reframes the mystery of why life is so unified today. Unity can come from success, not from exclusivity. If one lineage developed better energy handling, better copying, or better containment, it could outcompete others and eventually dominate the planet. The traces of lost beginnings would be subtle, because geology erases and because later life would recycle the same environments. The thought remains thrilling. Earth may have tried many times to become alive. The oldest evidence of life is older than most visible land on Earth. The Earth was active for a long time before humans came along, and that activity erases its own past. Mountains rise, then grind down. Ocean crust forms, then sinks back into the mantle. That is why the earliest traces of biology are so hard to find. Scientists look for hints that chemistry was shaped by living processes, even when obvious fossils are missing. One approach is to study how carbon appears in ancient rocks. Life tends to prefer lighter forms of carbon, and that preference can leave a recognizable pattern. The most ancient candidate signals come from rocks that have survived immense pressure and heat, which makes interpretation difficult. Still, the idea is staggering. If those signals truly reflect biology, then life was already present while Earth's surface was still being rewritten. Our planet may have hosted living chemistry remarkably early, despite impacts and a restless crust. The term quickly in geology still means tens or hundreds of millions of years. Human intuition struggles with geological time because our lives are short and our histories are recent. When scientists say life emerged quickly, they might still be describing an interval long enough for mountains to rise and vanish and for oceans to reshape coastlines many times over. This matters because it changes how you picture the first steps. You do not need a single miraculous afternoon. You have a vast span of repeated cycles, countless storms, endless tides, and uncountable chemical trials. Each day can be ordinary, yet the accumulation can be extraordinary. This kind of time also allows failure to be part of the process. Many chemical pathways can start and stop. Many protosystems can appear and vanish. Over long spans, rare successes become inevitable, because the planet keeps rolling the dice. Geological quickly is not rushed. It is persistent. It is the patience of a world that keeps offering chances, quietly, over and over, until something finally takes hold and begins to evolve. Every breath you take echoes an ancient microbial innovation called respiration. Breathing feels simple, but it rests on a clever trick that microbes pioneered. Respiration is about moving electrons through a chain of helpers, step by step, to harvest energy efficiently. Instead of burning fuel in one burst, cells use a controlled cascade that captures energy in a usable form. Long before oxygen filled the air, microbes likely used other substances as the final electron sink, such as nitrate, sulfate, or iron compounds. Later, when oxygen became available, it offered a powerful new option because it pulls electrons strongly. That made energy yields higher, and it reshaped what life could do. Inside your cells, mitochondria still run this ancient style of chemistry. They build an electrical gradient across a membrane, then tap it like a tiny turbine. So each calm inhale links you to deep time, to microbial experiments that discovered how to turn chemistry into steady power. Early life probably swapped genes freely, like a global experiment. Before evolution settled into stable lineages, genetic material may have moved sideways as easily as it moved from parent to offspring. In microbial life today, gene swapping can happen through viruses, through direct exchange, or through uptake of DNA from the environment. If early cells were simpler and lived in dense communities, those routes could have been even more common. That would make early evolution fast and inventive. A useful innovation, such as a better way to harvest energy or tolerate heat, could spread through a population without waiting for generations of descent. It also changes how we imagine the earliest family tree. Instead of clean branches, you get braided streams that join and separate. This idea helps explain why tracing deep ancestry is difficult, because the oldest signals may be mosaics. Yet it also offers a hopeful message. Life may have learned by sharing, not just by competing. The early biosphere could have been a planet-wide workshop, trading successful tricks across a microbial world. Horizontal gene transfer still reshapes microbes in real time. Even now, evolution can move at the speed of sharing. Bacteria can acquire entire sets of genes that grant new abilities, like using a new food source or resisting toxins. This is not a slow change of one mutation at a time. It can be a sudden upgrade, more like installing a new toolkit. In hospitals, agriculture, and natural ecosystems, this kind of transfer can change microbial communities rapidly. It also reveals something fundamental about life's origins. Genetic information is not always locked inside a single lineage. It can behave like a traveling resource. That makes early evolution easier to imagine, because the first useful genetic elements could spread through communities and become refined in many hosts at once. It also complicates the search for the earliest history, because gene transfer can blur the signal of direct descent. Still, the phenomenon is a living demonstration that life is not only about inheritance through family lines. It is also about exchange across neighbors, which may have been crucial in life's earliest chapters. The first living systems probably started without any cells at all. Cells feel essential now, but early evolution may have started in a looser way. Imagine chemical reactions happening on a mineral surface or inside tiny rock pores, where certain molecules keep getting remade. If a set of reactions helps create the ingredients it needs, it can persist longer than random chemistry. Add variation, add competition for resources, and selection can begin without a membrane in sight. Some researchers picture small strands of information carrying molecules like early RNA, sharing space with simple catalysts. Others imagine networks that cycle and grow because energy keeps flowing through them. Only later would a true boundary become useful, because it would keep helpful combinations together. In that view, cells are not the first chapter. They are an upgrade that helped successful chemistry become a stable lineage. Some modern microbes live on chemicals, not sunlight, like Earth's earliest life. There are places on Earth where sunlight never reaches, yet ecosystems still thrive. At deep-sea vents and within rocks, some microbes make a living by tapping chemical energy. They may eat hydrogen, sulfur compounds, or methane. They may use carbon dioxide as their carbon source, building their bodies from what seems like thin air. These lifestyles matter for origins because they show life does not need green plants to get started. It can run on chemistry alone. In some vent regions, microbes form the base of entire food webs, supporting worms, clams, and other animals that rely on those microbes. That is a reminder that food can mean electrons and reactions, not just leaves and sugars. If early Earth offered steady chemical fuel, life could have taken hold in the dark long before skies cleared. Life may have begun when minerals acted like tiny chemical batteries. Picture an early shoreline or a seafloor crack where fresh, hot fluids met colder seawater. In that meeting place, certain minerals can separate charges and shuffle electrons. That creates a natural voltage, like a battery you do not need to build. Rusty iron minerals and sulfur-rich rocks can help push carbon dioxide toward more complex molecules. The most intriguing part is that these mineral surfaces are not smooth. They are full of pores and channels which can hold onto ingredients and keep them close long enough to react. Over time, a network of reactions can start to feed on the energy difference around it. That is the kind of steady push chemistry needs before any genes exist to guide the work. Your DNA is built from atoms forged inside long-dead stars. Hydrogen and most helium were born in the early universe, but the heavier atoms in your body took a more dramatic route. Inside stars, intense heat and pressure fuse simpler elements into new ones. Later, when some stars swell and shed their outer layers, or when massive stars explode, those newly made atoms scatter into space. Carbon, oxygen, nitrogen, phosphorus, and iron can ride outward in dust and gas, mixing into future clouds that become new stars and planets. Earth formed from that recycled material, and so did the molecules that eventually learned to copy themselves. When you think about origins, it helps to zoom out. The story begins long before Earth had oceans. It begins with cosmic furnaces and a long chain of mixing and gathering that made chemistry on this planet possible. A simple lipid bubble can form by itself in plain water. Some molecules have a split personality. One end likes water, the other end avoids it. Drop enough of these into water, and they tend to assemble on their own. They can form spheres with a thin skin, creating an inside and an outside in a single step. That boundary matters because it makes a private space where chemistry can build momentum. Early bubble walls were likely leaky, which is a feature, not a flaw. Small nutrients can slip in, while larger molecules can stay put. Even better, these bubbles can grow if more fatty molecules drift into them. Under the right conditions, physical stress can pinch them into smaller bubbles. You do not need a blueprint to get this behavior. You need the right kinds of molecules and a world that keeps stirring, drying, wetting, and mixing. The same molecule can be both food and a building block. In early chemistry, a line between eating and building can blur. A small molecule might release energy when it reacts, and that same reaction can produce pieces useful for construction. In modern cells, this principle shows up everywhere. Simple compounds feed pathways that make sugars, amino acids, and nucleic acid components. For origins, that dual role is exciting because it means you do not need separate systems for energy and materials at the start. A molecule drifting in from the environment could power a reaction network while also becoming part of the growing structure. This helps explain how complexity can rise without careful planning. If useful chemistry makes more of itself and also makes sturdier products, it can gain a foothold. Over time, what began as opportunistic reactions can become organized cycles, and eventually those cycles can support inheritance. Life's chemistry may have begun as a messy mixture, not a perfect recipe. It is tempting to picture a clean, elegant pathway that leads straight to life. Real chemistry is rarely so tidy. Early Earth likely offered a cluttered soup of molecules made by heat, lightning, rock reactions, and incoming space material. Many of those molecules would interfere with each other. Some would gum up reactions. Some would break down quickly. Yet messiness can be an advantage because it creates a huge library of possibilities. If a surface, a pool, or a pore system keeps concentrating and filtering that mixture, certain reactions can start to dominate. Over repeated cycles of change, the chemistry that persists becomes the chemistry that matters. That is selection in a chemical form. Instead of a single lucky molecule, you might get a whole shifting community of reactions. From that crowd, patterns can emerge, and those patterns can slowly become more lifelike. Lightning can stick simple gases into amino acids in the lab. A high-energy spark can rip stable molecules apart and leave behind reactive fragments that quickly recombine. When those fragments cool and meet water, some of the products can become amino acids, which are key building blocks for proteins. The power of this idea is not that lightning is the only route, but that it proves something important. Complex, life-related molecules do not require living cells to appear. Energy plus the right ingredients can be enough to start. On early Earth, sparks would have been only one source of energy in a noisy environment. Ultraviolet light, volcanic heat, and shock waves from impacts can also drive rearrangements. Each pathway makes a different blend of compounds, and blends create options. The deeper question becomes, which environments could collect those options, protect them, and give them repeated chances to link into larger structures? Hydrothermal vents create natural chemical gradients that can power reactions. At the seafloor, hot fluids rise from cracks in the crust, carrying dissolved chemicals from deep rock. When that fluid meets cold seawater, sharp differences appear across tiny distances. Temperature shifts, acidity shifts, and the mix of available chemicals shifts as well. Those differences matter because they provide direction. Chemistry is more likely to run when there is an imbalance to spend. In vent regions, reactions can be pushed forward again and again, not by rare luck, but by continuous flow. Many origin ideas focus on this persistence. A vent field can run experiments for thousands of years without stopping, constantly delivering fresh reactants while carrying away waste. It is a setting where energy is not a sudden flash. It is a steady pressure that can sculpt pathways over time, gradually favoring reactions that reinforce themselves. Some vents build porous rock chimneys that act like microscopic rooms. Mineral-rich fluids can precipitate solids as they mix with seawater, building tower-like structures that grow upward from the sea floor. Inside those structures are countless pores and thin channels, which can trap molecules instead of letting them drift away. Concentration is a quiet superpower in origins research. When ingredients are held close, reactions that are unlikely in open water can become routine. The walls can also offer catalytic surfaces that encourage certain transformations. Imagine millions of tiny chambers in one chimney, each chamber running a slightly different chemical trial. Most trials lead nowhere, but a few may produce compounds that feed other reactions nearby. Over time, successful chains can spread through the connected pore network. A chimney is not alive, yet it provides the kind of organized space early chemistry struggles to find. It is structure before biology and a possible stepping stone toward heredity. Warm metal ponds could concentrate molecules as water repeatedly evaporated. In a wide ocean, useful ingredients can be scattered so thinly that complex reactions stall. A small pond behaves differently because it changes with the weather. Sun warms it, rain refills it, and dry periods lower the water level until dissolved molecules become crowded together. Crowding increases the odds that two compatible molecules meet and react, especially when the shoreline leaves behind sticky films and damp sediments. When water returns, products can dissolve, mix, and travel to new microspots, which sets up the next cycle. Repetition can do what a single moment cannot. It can select for chemistry that survives drying, survives rewetting, and keeps building. A pond also offers variety over short distances. One edge may be salty, another may be mineral-rich, another may be shaded. That patchwork lets many chemical stories run in parallel, and some stories can join into longer ones. Freezing can also concentrate chemicals by squeezing them between ice crystals. When water freezes, the crystal lattice tends to exclude many dissolved substances. Those substances get pushed into tiny unfrozen channels that remain liquid even in a mostly icy environment. In those narrow spaces, concentrations can rise dramatically, and molecules that rarely meet in dilute water can suddenly collide often. Cold also slows many destructive reactions, which can help fragile compounds persist long enough to participate in something more complex. This creates a useful combination: concentration plus protection. Seasonal environments are especially intriguing because they add rhythm. Freeze concentrates and preserves. Thaw mixes and redistributes. Then freezing happens again. Each cycle gives chemistry another chance to link, rearrange, and build. It also offers gentle sorting, because some molecules prefer the icy interface, while others remain in the liquid pockets. In that way, winter conditions can act like a natural laboratory, nudging mixtures toward organization without any guiding hand. Meteorites carry amino acids, showing complex chemistry can form in space. Some meteorites are ancient time capsules, formed before Earth and preserved in the cold of space for billions of years. In certain carbon-rich specimens, scientists have found a surprising variety of amino acids, including types that biology barely uses. That diversity is a clue that the molecules formed through non-living chemistry, then rode inside rock until the meteorite fell to Earth. The implication is thrilling. Early Earth may have received a steady drizzle of organic ingredients, not just whatever the planet could manufacture locally. Those ingredients could land in oceans, lakes, hot springs, or wet sediments and join the chemistry already in progress. Even if space delivered only small amounts repeated over long time can matter. It widens the stage for life's beginnings. It suggests that planets may start with help, carrying pre-made molecular pieces from the larger solar system and perhaps beyond. Comets contain organic molecules, including ingredients tied to sugars. Comets are icy visitors that preserve early solar system material in deep cold storage. When studied up close, they reveal carbon-based compounds that formed on frozen grains, exposed to radiation over long stretches of time. Among those compounds are small reactive molecules that can participate in pathways toward sugars, such as simple aldehydes. That matters because sugars are central to biology. They store energy, They shape cell surfaces, and they form the backbone of RNA and DNA. A comet does not need to deliver finished sugars to be important. It only needs to deliver useful starting pieces that are ready to react. In the young solar system, comet impacts and close passes were likely more frequent than they are now. Each encounter could add new chemical possibilities to Earth's surface. In that view, the early planet was not working alone. It was part of a larger chemical neighborhood, trading materials across space. Interstellar dust grains can help assemble organics in deep space cold. Between the stars, gas is thin and temperatures are extremely low, yet chemistry still finds a way to move forward. Tiny dust grains provide surfaces where atoms and simple molecules can stick instead of drifting apart. Over time, icy layers build up on those grains, and radiation can rearrange what is trapped in the ice, producing more complex organics. This means some life-relevant chemistry can begin long before planets form. When a new star is born, the surrounding disk can inherit that processed dust, carrying it into comets, asteroids, and eventually young worlds. The story of origins then gains a quiet prologue. Earth may have started with ingredients that already had a chemical history, shaped by cold, time, and starlight in deep space. It also strengthens the hope that life's starting materials are common. If dust grains can do this almost anywhere, then many planets may begin with similar molecular opportunities. Clay surfaces can help align molecules so they react more easily. In open water, molecules drift and spin, and productive encounters can be rare. A mineral surface changes the game by holding molecules close and, in some cases, lining them up in helpful orientations. Many clays have layered structures and charged sites that attract organic compounds. That attraction can concentrate ingredients and keep them from washing away. The surface can also create micro-conditions that differ from the surrounding water, which can favor reactions that are otherwise difficult. This becomes especially interesting for forming longer chains, because building polymers often requires repeated, well-timed connections. A clay-rich sediment bed can act like a scaffold, offering countless attachment points where small units can gather and link. Different clay types can prefer different molecules, which adds a kind of sorting. Over long periods, such sorting could help reactions become less random and more pathway-like, which is exactly what early chemistry needs before heredity becomes reliable. Certain minerals can favor one molecular handedness over the other. Imagine two gloves that look alike, yet only one fits your hand. Many important molecules come in these mirror image forms, and early Earth had to pick a side somehow. Some crystal surfaces are not symmetrical, so the tiny grooves and charged spots can interact more strongly with one hand than the other. When a mixture repeatedly washes over those surfaces, a small preference can grow into a noticeable imbalance. It is like a coin that is only slightly weighted, flipped enough times to reveal its bias. This matters because once a system begins using mostly one-handed form, copying and building become easier. Parts fit better. Chains form more consistently. Researchers test minerals such as quartz and certain clays to see how strong these effects can be under realistic conditions. A quiet, persistent nudge from geology may have helped chemistry choose a direction. Life uses mostly left-handed amino acids, and we still debate why. Biology is picky in a surprising way. The amino acids in your proteins are almost all one-handed form, and the opposite form is mostly absent from life's main machinery. That choice is not required by basic chemistry. Both forms can form without life. The mystery is how early processes broke the tie. One possibility is that a tiny initial imbalance was amplified by repeated cycles of building and breaking. Another idea points to polarized light in space, which can destroy one-handed form a bit faster than the other in certain conditions. There is also the possibility that chance played a role, and once a particular handedness gained a foothold, evolution locked it in because switching later would be catastrophic. Whatever the cause, the consequence is huge. This single preference shapes how proteins fold, how enzymes work, and how living chemistry stays coherent instead of chaotic. RNA can store information and also act as a catalyst. It is hard to imagine a simpler bridge between chemistry and biology. A strand of RNA can hold a sequence which works like a message about how to build. Yet RNA can also fold into shapes that do work, including speeding up reactions. That means one kind of molecule can both remember and act. In modern life, RNA already performs a wide range of jobs, from carrying instructions to helping control genes like dimmer switches. In Origins research, this dual ability is captivating because it offers a path that does not require a full toolkit from the start. You can begin with one versatile player. If some RNA-like molecules formed in an environment that allowed them to persist, certain sequences could have produced shapes that helped them last longer, spread faster. Selection could then favor the strands that not only exist, but also improve their own odds of continuing. Some RNA molecules can copy parts of themselves under lab conditions. This is where the story starts to feel almost alive. In carefully designed experiments, researchers have created RNA strands that can guide the joining of smaller pieces into a complementary copy. It is not effortless, and it is not yet the open-ended replication that a living cell achieves. Still, it shows that the gap between chemistry and heredity is not an infinite chasm. The copied strand can carry information that influences what happens next, which is the seed of evolution. The struggle is accuracy. If copying is too sloppy, information dissolves into noise. If copying is too slow, the system cannot compete with breakdown. That tension pushes scientists to explore conditions that help, such as cycles of warming and cooling, or the presence of metal ions that stabilize structures. Each result is a glimpse of a possible early world where molecules began to make family lines. A world of RNA could have preceded DNA, proteins, and cells. Picture a time when the lead actor was not DNA and not proteins, but a flexible molecule that could do a bit of both jobs. In this scenario, RNA-like chains carried the instructions for their own continuation, and some of those chains folded into shapes that helped reactions along grow. Over time, networks of RNA could have become more efficient, more diverse, and more competitive. Then, as chemistry improved, new specialists could appear. Proteins can become far more powerful catalysts, so they would be an advantage once they could be made reliably. DNA is sturdier for long-term storage, so it would be a better archive once the system was sophisticated enough to maintain it. The beauty of this idea is its gradualism. It does not require every modern feature at the start. It offers a sequence of upgrades, each one making the next more likely, until cells and genomes become the stable home for an evolving lineage. DNA is more stable than RNA, which may explain why it took over. If you want to keep a message safe, you do not write it on paper that dissolves in rain. RNA is chemically reactive, and its backbone is easier to break. That reactivity can be useful early on, because flexibility allows experimentation. Yet, as heredity becomes important, fragility becomes a liability. DNA solves that problem with a slightly different chemistry and a double-stranded design that provides backup. If one strand is damaged, the other can guide repair. That makes long genomes possible, which opens the door to more complex organisms and more elaborate regulation. The transition would not need to be sudden. A system could use RNA for many tasks, while gradually adopting DNA for storage. Even today, life preserves echoes of that history. Many viruses still use RNA genomes, and your own cells use RNA as the messenger and the worker. Stability was not just a convenience. It may have been the turning point that allowed evolution to keep its hard-won information. Proteins are powerful catalysts, but they need information to be built. Proteins are like custom tools. One shape grips a molecule. Another shape twists it. Another shape snaps bonds and makes new ones. With enough variety, proteins can run nearly every reaction a cell needs. The catch is that proteins do not copy themselves. They must be assembled in a precise order, and that order is information. That creates a classic puzzle in origins research. How do you get excellent catalysts before you have a reliable instruction system? And how do you get a reliable instruction system before you have excellent prol? Catalysts? One appealing answer is teamwork. Simpler catalysts could have existed first, perhaps small molecules or mineral surfaces, and they could support the emergence of information carrying polymers. Later, those polymers could begin encoding proteins, which then took over much of the catalytic workload. It is less like a single invention and more like a partnership that gradually deepened until modern biology emerged. Ribosomes are part RNA, hinting at an ancient RNA-based past. Every time a cell makes a protein, it relies on a machine that feels like a fossil in motion. The ribosome is built from both protein and RNA, yet the core job of linking amino acids is carried out by RNA at its heart. That detail matters because it suggests an older era when RNA did more of the heavy lifting. Over evolutionary time, proteins may have been added around that core, like protective scaffolding, improving speed and control while keeping the ancient truths. Center intact. The ribosome is also remarkably universal across life, which implies it appeared early and became too important to replace. When you listen to this story, it helps to imagine billions of years of continuity. A molecule that once helped chemistry become heredity is still operating inside you right now. It is not a metaphor. It is a working relic, translating genetic messages into living structure, one link at a time. Modern viruses blur the line between chemistry and biology. A virus can be startlingly minimal. It carries genetic instructions, yet it cannot reproduce on its own. It must enter a host cell and borrow the cell's machinery to make copies. That in-between status makes viruses fascinating for origins, even though they are not usually considered ancestors of the first life. They show how little you can have and still participate in evolution. A virus genome can mutate, compete, and adapt with fierce speed. Yet the virus itself is not a self-sustaining system. Some scientists think viruses may preserve echoes of ancient genetic elements, pieces of early evolution that learned to travel between cells. Others see viruses as later inventions that emerged after cells existed, as genetic escape artists. Either way, they force a deeper question. What should count as alive? If evolution can act on something that is only partly independent, then the border between life and non-life becomes softer, and the origin story gains proper-en. More possible stepping stones. Some researchers think metabolism came first, before genetic information. Instead of starting with a molecule that copies itself, this view starts with a network that keeps going. Imagine a set of reactions that feed one another, powered by energy from the environment. If the network uses available chemicals and produces intermediates that help it persist, it can become a kind of chemical ecosystem. Over time, such networks could become more structured, especially if they take place in a setting that provides steady inputs and removes waste. Only later would an information system become valuable. because it could stabilize the best-performing networks and help them spread into new places. This approach shifts the drama. It says, life did not begin as a perfect replicator. It began as organized flow. Then heredity arrived as an upgrade, a way to remember successful chemistry. The appeal is that many core metabolic pathways in modern cells feel deeply rooted, as if they might trace back to the earliest energetic scaffolding that made living grow. Persistence possible. Metabolism-first ideas focus on reaction networks that self-sustain. Instead of starting with a molecule that copies itself, this approach starts with a cycle that keeps its footing. Picture a set of reactions where one product becomes the next reaction's fuel, and the chain loops back on itself. If the environment keeps supplying a few simple inputs, the loop can persist like a flame that keeps finding oxygen. The most exciting part is that such networks can show a kind of primitive selection. Loops that run faster can hog resources. Loops that tolerate interruptions can outlast dry spells or temperature swings. Some networks may even generate side products that stabilize the loop, like making a helper molecule that improves a key step. Over a long time, the chemistry that survives is the chemistry that becomes common. In this view, inheritance arrives later as a way to preserve the best loops, not as the opening act. Iron and sulfur minerals can catalyze reactions like those in cells. Some of the most ancient-looking chemistry in biology relies on metals, and iron and sulfur are stars of that story. In many enzymes today, clusters of iron and sulfur sit at the active center, passing electrons and enabling reactions that would otherwise crawl. On the early Earth, iron and sulfur minerals were widespread, especially in volcanic and hydrothermal settings. That raises a thrilling possibility. Before enzymes existed, mineral surfaces could have done a rough version of the same job. They can help move electrons, stabilize short-lived intermediates, and encourage carbon to link into larger structures. This creates a bridge between geology and biology that feels almost intimate. The living world may have borrowed tricks from rock chemistry, then refined them into protein machines. When you look at an iron-sulfur cluster inside a modern cell, you may be seeing a polished echo of ancient minerals that once worked in the open. Carbon dioxide can be turned into organics using chemical energy. Carbon dioxide seems like an endpoint, yet it can also be a beginning. With the right energy source, it can be pushed into more complex carbon compounds, which is exactly what early life would have needed. In certain geochemical settings, hydrogen is available, and hydrogen can act like a chemical battery waiting to be discharged. Combine carbon dioxide with hydrogen, add helpful mineral catalysts, and you can begin building small organics that resemble the starting pieces of tau. Metabolism. This matters because it suggests a direct route from simple planetary gases to richer chemistry, without requiring a pre-existing organism to do the work. It also reframes the atmosphere as a pantry, not just a backdrop. Early Earth could continuously supply carbon dioxide, while Earth's interior supplied energy-rich chemicals. When those streams met, the planet may have been quietly manufacturing the kinds of molecules that later became food, structure, and information. Life needs compartments, and membranes can form without any genes. To evolve, chemistry needs boundaries. Without a boundary, useful products drift away, and successes cannot stay together long enough to improve. The remarkable thing is that boundaries do not require a genetic blueprint. Certain simple molecules naturally assemble into sheets and spheres in water, creating an inside that is different from the outside. Once you have an inside, you can begin to build a local chemistry, one that can be fed, protected, and modified by conditions at the surface. A compartment also turns selection into something physical. If one bubble holds a chemistry that helps it grow or persist, it can become more common than its neighbors. Early compartments were likely imperfect and leaky, which can actually help because nutrients can enter without complicated transport systems. In Origin's research, compartments are not a decorative detail. They are a crucial step toward making chemistry behave like a lineage instead of a scattered crowd. Fatty acid vesicles can grow by absorbing molecules from their surroundings. A primitive membrane does not need pumps or proteins to expand. If the surrounding water contains more fatty molecules, a vesicle can simply take them in. The new molecules slip into the membrane like extra bricks sliding into a wall, and the bubble surface area increases. Growth is not only about getting bigger, it changes tension, shape, and stability, which can influence what happens to any chemistry inside. A growing vesicle can also outcompete others by soaking up the available membrane building material, leaving neighbors with less. That sounds almost biological, yet it can happen through basic physics and chemistry. This is one reason protocell models are so captivating. You can get something that looks like a lifelike behavior, feeding and growing, without needing DNA or enzymes. It is a plausible stepping stone, where the first winners are simply the containers that grow best under a planet's changing conditions. Vesicles can divide when shaken or pushed through tiny pores. Division does not have to begin with a complex cellular machine. If a membrane bubble grows large and gets stressed, it can split. Physical forces can do the job. Turbulent water, wave action, flowing through narrow mineral channels, or being squeezed between grains of sediment can pinch a vesicle into two. What makes this exciting is the possibility of a full cycle. Growth pulls in new membrane material. Then, environmental motion divides the container. If the internal contents are shared between the daughters, you suddenly have something like reproduction, even if it is messy. Some daughter vesicles will inherit more helpful chemistry than others, and that creates differences that selection can act on. This kind of division is not precise, and early evolution likely was not precise. It may have been a long era of imperfect copying, where persistence mattered more than perfection. Over time, any chemistry that helped vesicles grow and survive would be favored, setting the stage for more controlled division later. Simple protocells can maintain internal chemistry different from the outside. Once you have a boundary, the inside can become its own small world. Even a leaky membrane can create differences because some molecules cross more easily than others. That means a protocell can concentrate certain ingredients while excluding others, which changes reaction rates and outcomes. Inside, molecules can bump into each other more often. Products can accumulate rather than being swept away. A protocell can also buffer its contents against sudden changes outside, like a brief shift in acidity or salt. This is where the story starts to feel truly alive. Not because it is conscious or complex, but because it has an identity. The internal chemistry becomes something that can persist through time, and the persistence is the raw material for evolution. If two protocells experience the same environment but maintain different internal mixtures, they will behave differently. Some will last longer. Some will grow faster. Those differences can be inherited in a loose way when protocells divide, and evolution can begin to sculpt the chemistry. Salt levels can make some primitive membranes fall apart or stabilize. Salt is not just seasoning. In early chemistry, it can decide whether a protocell survives the day. Many simple membranes are made from fatty acids, and fatty acids carry charge that interacts strongly with ions in the water. In one salt condition, the membrane can stay flexible and intact. In another, it can clump, leak, or collapse. This is fascinating because it ties origins to geography. A shoreline pool, a rain-fed lake, and a deep ocean basin can present very different ionic environments. Small shifts could determine which membrane chemistries are even possible. Salt can also influence what enters a protocell. Some ions can neutralize charges and help certain molecules cross. Others can block entry or trigger precipitation. The result is that a protocell is not only competing with other protocells, it is also negotiating with its environment at every moment. In Origin's research, salt becomes a selective pressure, guiding which early compartments can persist long enough to matter. Early oceans may have been too salty for some protocell models. Many protocell experiments work beautifully in fresh water, then struggle when the salt rises. That matters because Earth's early ocean was not a gentle, controlled beaker. It was a chemically complex and salty world, and salt can destabilize fatty acid membranes by disrupting their delicate balance of charge and packing. This challenge has pushed researchers to think carefully about where early compartments could have formed. It also encourages creative solutions. Some membrane mixtures are more robust, especially when combined with additional amphiphiles that strengthen the structure. Some settings could offer lower salinity pockets, such as near river inflows, in volcanic land pools, or in transient environments after rainfall. The larger lesson is important. Not every appealing Origin idea can work everywhere on the planet. The environment selects, and it selects early. If a protocell model cannot tolerate realistic salt levels, it either needs a special niche or it needs a different kind of boundary. That tension is not a flaw in the science. It is the science, forcing ideas to face the real Earth. Freshwater environments might have been crucial for the first membranes. Freshwater offers something the ocean often does not, which is gentler chemistry for fragile early compartments. Lower salt can allow fatty acid membranes to form, remain stable, and keep useful molecules from leaking out too quickly. Freshwater settings also tend to be dynamic. Springs, streams, puddles, and lakes rise and fall, concentrate and dilute, warm and cool. Those changes can drive repeated cycles that help chemistry build complexity. A small lake edge can gather organics from runoff, ash, and weathered rock. It can also provide surfaces like clays and sediments that concentrate molecules and encourage reactions. This is why many Origin scenarios pay close attention to land-based water. It is not because the ocean is unimportant. It is because a freshwater niche might offer the right balance of stability and change, giving protocells time to form, grow, divide, and experiment. If membranes were a key step, freshwater could have been the cradle that made them practical. Deep-sea vent models avoid salt problems using mineral pores instead. In the open ocean, high salt can sabotage fragile early membranes. Some Origin scenarios sidestep that by giving chemistry a different kind of container. Vent minerals can precipitate into sponge-like rock filled with microscopic channels. Those channels can trap molecules the way a reef traps water, creating pockets where ingredients stay close long enough to react. Instead of a fatty boundary, the walls are stone, and the separation is physical. Flow through the pores can bring in fresh reactants and carry away waste, which keeps reactions from stalling. The pool walls can also provide catalytic surfaces that nudge molecules into new arrangements. Over time, a network of neighboring pores could behave like a connected neighborhood of microreactors. If one pocket develops a useful cycle, its products can drift into the next pocket and extend the chemistry. It is a compelling picture of lifelike organization before true cells existed. Natural pH gradients across rocks resemble the ways cells make energy. Some vent systems place alkaline fluids beside more acidic seawater, and the boundary can be sharp across a thin mineral wall. That creates a pH gradient, which is a difference in the concentration of hydrogen ions. Modern cells use a similar kind of difference to power themselves. They maintain an imbalance across a membrane then let ions flow back through molecular machines that capture the energy. In a rocky setting, the wall is mineral, not lipid, yet the principle is familiar. Differences want to equalize, and that drive can be harvested to push chemistry forward. A pH gradient can influence which molecules form, which reactions run, and which structures remain stable. It can also create directional flow of charge, which matters for building complex organics from simple carbon sources. When you hear that life runs on gradients, it sounds abstract. In a vent setting, it becomes tangible. It is Earth's own battery, built from rock and seawater. ATP is life's energy currency, and it acts like a charged spring. Cells need a portable way to capture energy and spend it precisely, reaction by reaction. ATP fills that role by storing energy in its phosphate bombs, ready to be released when a cell needs to build, move, or repair. Think of it as a wound spring that can be snapped open on demand. What makes ATP so fascinating for Origins is that it sits at the crossroads of many processes. It can pay for making proteins. It can pay for copying genetic material. It can pay for moving ions and molecules across membranes. That universality suggests early evolution found a solution that was versatile and then refused to let it go. The harder question is how a system first began making and using such an energy token before sophisticated enzymes existed. Researchers look for simpler molecules that could have played a similar role at first, then gradually yielded to ATP as biochemistry matured. Many cells still harvest energy using proton gradients across membranes. Inside your cells, energy production is not mainly a little flame. It is an electrical and chemical imbalance across a membrane. Protons are pushed to one side, building a gradient like water held behind a dam. When protons flow back through a rotary enzyme, the enzyme turns that flow into ATP. This strategy is stunning because it is both delicate and powerful, and it appears across huge branches of life. It hints at deep ancestry, as if early evolution discovered a winning design and built upon it for billions of years. Gradients are attractive for origin theories because nature can create them without biology. Vents can do it. Rock chemistry can do it. Even simple membranes can hold a difference for a while. Once a protocell could maintain any gradient at all, it would gain a steady source of usable energy, which could support longer chains, better catalysts, eventually more reliable inheritance. The universal genetic code is shared by almost all known life. Every cell translates genetic instructions into proteins using the same basic dictionary, where three-letter nucleotide words map to amino acids. That shared system is so widespread that it is one of the strongest clues that life on Earth is deeply related. It is also a strange kind of fossil. The code is not the only possible code. Many alternatives could work in principle, yet evolution settled on one and then built everything else on top of it. Once the code became central, changing it would be risky because every protein would be affected at once. That makes the code feel like an early lock-in event, a choice made when life was still young and flexible. The rare exceptions and small variations we see today are interesting precisely because they are limited. They show that the code can bend a little, but it usually does not break. That stubborn consistency is part of what makes origins feel real rather than mythical. That shared code suggests all life today has a common ancestor. When you see the same translation system in bacteria, plants, and animals, it implies a single deep lineage behind today's diversity. This does not mean evolution had a neat starting point with one perfect cell. It means that, at some stage, one family of early life became so successful that its descendants filled the planet and outlasted competitors. The common ancestor idea is powerful because it narrows the mystery. Whatever life's first steps were, they led into a lineage that learned how to store information, translate it into working chemistry, and pass improvements forward. It also means you carry an ancient inheritance that predates bones, forests, and even oxygen-rich skies. The details of that ancestor remain hidden because early life left few clear fossils, and because gene swapping can blur family trees. Still, the shared code is a signature that points backward, toward a single root somewhere in deep time. The last universal common ancestor was likely a community, not one cell. It is tempting to imagine one tiny organism standing alone at the dawn of life. Many scientists suspect the reality was more like a bustling microbial neighborhood. Early cells may have been simpler and leakier, and they may have shared useful molecules with their surroundings. If genes moved easily between neighbors, then innovation could spread quickly, and the ancestor becomes less like a single individual and more like a population disease. That collectively explored solutions. This community view also helps explain why some core features are universal while other features vary widely. A shared pool of genetic parts could be mixed and matched, with successful combinations persisting. In that era, boundaries between lineages may have been fuzzy, closer to a network than a tree. Over time, as membranes improved and genomes became more coordinated, lineages could become more distinct. The last universal common ancestor, in this picture, is a shared chapter of communal evolution rather than one lonely cell. The earliest ecosystems were microbial mats, layered like living carpets. Long before animals swam or plants rooted into soil, microbes formed dense communities across shallow waters and wet surfaces. A microbial mat can be layered, with different organisms living in different zones based on light, oxygen, and chemistry. The top layer may catch sunlight. Lower layers may rely on chemical reactions that use the byproducts from above. This creates a miniature ecosystem in a few centimeters, with gradients that change over the course of a day. Mats can also trap sediments, bind grains together, and gradually build structures that can become rock-like over time. What makes mats so captivating is their cooperative architecture. They show how simple organisms can build complex habitats just by living side by side. For origins and early evolution, mats offer a plausible stage where chemistry, energy capture, and recycling could intensify. They concentrate life, they create microenvironments, and they preserve traces that can survive into the geological record. Stromatolites are rock-like structures built by microbes over time. Layer by layer, microbial communities can trap sediment and bind tiny grains together, slowly making a structure that looks more like stone than life. The builders are often microbes that live in dense sheets, where the surface is active and the deeper layers are sheltered. As water flows over them, grains settle and stick. Over days and seasons, those grains become part of the mat's growing architecture. The result can rise into domes, ridges, or columns, depending on currents and sunlight. What makes this astonishing is the timescale. A living film can create a lasting geological signature, one quiet day at a time. These structures show that early life did not need claws or shells to leave a mark. It could shape landscapes with patience, turning metabolism into architecture and turning a shoreline into a record keeper. Some stromatolites are living today, offering a window into deep time. In a few places on Earth, stromatolite communities still grow, which means you can stand beside a modern ecosystem that resembles some of the planet's earliest ones. These living sites are rare because most environments are now crowded with grazers and competing organisms that can break mats apart. Where conditions are challenging, such as very salty lagoons, microbial layers can still dominate. That rarity is part of the appeal. It highlights how different the early world was, when microbes had vast space and little interference from larger life. Studying living stromatolites helps scientists connect biology to geology in a direct way. You can observe how layers form, how sediment becomes trapped, and how microbial activity changes the chemistry around it. It is not a perfect time machine, because today's Earth is different. Still, it is a tangible reminder that ancient styles of life never fully vanished. They persisted in pockets, quietly continuing a tradition older than animals. Photosynthesis began without oxygen, using other chemicals as electron sources. The first photosynthesizers likely did not spit water, and they did not release oxygen as a byproduct. Instead, they used sunlight to pull electrons from other substances, such as hydrogen sulfide or dissolved iron compounds. That means early photosynthesis could operate in environments that would seem strange today, including shallow waters with the smell of sulfur or seas rich in qua. Reactive metals. This matters because it shows sunlight could become biologically useful before the planet's atmosphere was transformed. It also suggests a gradual path toward the powerful oxygen-producing version that later emerged. Early photosynthesis would have changed local chemistry, building organic matter and shifting which compounds were available nearby. In a microbial mat, different layers could cooperate, with one group's waste becoming another group's resource. That kind of recycling creates stability, and stability gives evolution room to experiment. Sunlight did not have to remake the whole world at once. It could begin as a local advantage, then spread. Oxygenic photosynthesis later changed the planet more than any volcano. When a lineage learned to use water as the electron source for photosynthesis, it unlocked an enormous supply of raw material, because water is everywhere. The byproduct was oxygen, and oxygen does not stay polite and local. It reacts, it accumulates, and it reshapes chemistry on a planetary scale. Over a long time, this innovation turned the atmosphere into something radically new and made entirely new kinds of metabolisms possible. It also opened the door to larger bodies, because oxygen allows cells to extract far more energy from food than many older pathways. That extra energy supports complexity, speed, and active lifestyles. The transformation did not happen overnight, and for a long while, oxygen was consumed by rocks and dissolved metals. Still, the direction was set. A microbial trick with sunlight became a global force, altering oceans, air, and the range of futures available to life. It is hard to name another natural process that redirected Earth so completely. The Great Oxidation Event filled oceans and air with reactive oxygen. At a certain point in deep time, oxygen began to rise beyond local pockets and become a major player in the wider environment. That shift changed ocean chemistry first, then gradually altered the atmosphere as well. Oxygen is eager to react, so its appearance meant many substances that were stable before could suddenly be transformed. Metals oxidized. New minerals formed. Old chemical pathways became harder to sustain. The event is famous because it marks a turning point when Earth's surface became less friendly to many older forms of life, yet more promising for others. It also created new ecological opportunities, because oxygen can serve as a powerful ingredient for energy harvesting. The planet did not simply gain a new gas. It gained a new kind of chemistry, one that made the environment more dynamic and, in many ways, more dangerous. Life had to adapt to this restless reactant, either by avoiding it, enduring it, or learning to use it. In that pressure, evolution found new directions. Oxygen was toxic to many early microbes, causing a global crisis. For organisms adapted to an oxygen-poor world, oxygen could be a chemical threat. It can form reactive byproducts that damage proteins, membranes, and genetic material. Early microbes that had never faced this challenge would have been vulnerable, especially in surface waters where oxygen first appeared. The crisis was not a single disaster with a clear ending. It was a long period of environmental change where familiar habitats became risky. Some lineages likely retreated into oxygen-free niches, such as deep sediments, hot springs, or the subsurface. Others survived by evolving defenses, including enzymes that neutralize harmful oxygen compounds. A smaller number learned to turn oxygen into an advantage, using it to extract more energy from food. That split is one reason oxygen feels so dramatic in the history of life. It acted like a new rule suddenly introduced to the planet, and every organism had to respond. Some lost ground. Some reinvented themselves. Some flourished in the New World. Earth's iron-rich seas rusted, creating banded iron formations. Before oxygen became abundant, dissolved iron could travel widely through seawater. Once oxygen began to appear in larger amounts, that iron had a new partner, and the reaction was dramatic. Oxygen combined with dissolved iron to form insoluble iron oxides, which settled out of the water like a slow planetary snowfall. Over a long time, layers of iron-rich minerals built up on the sea floor, often alternating with other sediments. These deposits became banded iron formations, and they are among the clearest geological signatures of changing ocean chemistry. What makes them so compelling is that they link life and rock in a visible way. Biological oxygen production helped trigger a chemical cascade that literally wrote itself into stone. In a sense, the oceans recorded their own transition from a metal-rich, oxygen-poor world to a more modern state. Each band is a whisper from ancient water saying, "Something changed here." And the change was big enough to leave a mark that still persists. Ozone formed from oxygen, shielding life from harsh ultraviolet light. Once enough oxygen existed high in the atmosphere, sunlight could break some oxygen molecules apart, and the fragments could recombine into ozone. Ozone is a thin layer by everyday standards, yet it has an outsized effect because it absorbs much of the most damaging ultraviolet radiation. That matters for origins and evolution because ultraviolet light can shred complex molecules, including the building blocks of genetic systems. With stronger shielding, surface environments become safer places for biology to spread and diversify. It also changes which strategies are favored. When radiation pressure drops, organisms can invest less in emergency repair and more in growth, interaction, and ecological complexity. This protection would not have been perfect, and it would have developed gradually. Still, it represents a kind of atmospheric kindness that arrived only after life had already transformed the planet. There is something beautifully circular about that. Life helped create oxygen. Oxygen helped create ozone. Ozone helped make the surface more livable for life. The planet and its biology began to co-design each other. Early continents were smaller, and oceans covered more of the planet. In the distant past, much of Earth's surface was water, and the land that did exist may have been more fragmented. Smaller continents mean different coastlines, different nutrient delivery, and different kinds of shallow environments where chemistry and biology can concentrate. Shorelines are special because they mix ingredients from rock and water, and they offer cycles of wetting and drying that can drive chemical change. With more ocean area, seafloor environments also become prominent, including volcanic ridges and hydrothermal systems that can feed chemical energy into surrounding water. A world dominated by oceans also changes climate patterns because water stores heat and moves it around the globe. This shapes where stable habitats can form and how often they are disrupted. When you picture life's early chapters, it helps to imagine a planet where land was rarer, Coastlines were constantly shifting, and shallow seas may have been widespread. Laboratories. The geography itself could have guided where early ecosystems took hold. Plate tectonics may have helped recycle nutrients needed for early life. Life needs more than carbon and water. It needs elements like phosphorus, sulfur, and nitrogen, plus trace metals that make crucial reactions possible. Plate tectonics can help keep these supplies moving by continually renewing the surface. Rocks are uplifted, weathered, carried to the sea, buried, transformed, and returned again through volcanic activity. That cycling can deliver fresh minerals to water, and it can create new habitats where chemistry is energetic and varied. Subduction zones and volcanic arcs can also release gases and nutrients that feed microbial communities. Even the formation of new ocean crust can influence ocean chemistry, which affects how nutrients are available and where they concentrate. The most intriguing thought is that tectonics may have stabilized life's long-term prospects. Without recycling, a planet can become chemically stagnant, with key elements locked away. With recycling, the environment stays inventive. It keeps offering new surfaces, new flows, and new gradients for life to explore. In a deep way, the moving crust may have been part of life's support system. Impacts were common, and some may have sterilized the surface briefly. Early Earth was pelted by leftover debris from solar system formation, and the largest impacts could have been truly world-shaking. A big strike can heat the air, ignite widespread fires, and drive conditions that are brutal for shallow habitats. In the most extreme cases, surface waters could temporarily become hostile, and any young ecosystems near the top would be at risk. Yet impacts are not only destruction. They also fracture crust, expose fresh minerals, and can create hot water circulation in crater zones that lasts long after the initial blast. That aftermath can become a new chemical setting, with heat, gradients, and newly available ingredients. In a strange way, a violent reset can also create new opportunities for chemistry to reorganize. Life's early path may have been shaped by interruptions, where survival depended on finding shelter and then taking advantage of new environments once the planet, quote, calmed. Deep habitats could have sheltered life through violent bombardment. A few meters of water or rock can buffer heat and radiation far better than open air. And deeper still, conditions change more slowly. Beneath the sea floor, pores and cracks can hold water and provide steady chemical energy from reactions between rock and fluid. During periods of frequent impacts, these deeper settings could remain comparatively stable while the surface was repeatedly disturbed. Pressure underground also keeps water liquid at temperatures that would boil at the surface, which widens the range of livable conditions. This shelter idea makes early survival feel less like a miracle and more like good geography. Life does not need the entire planet to be calm. It needs one dependable niche that lasts long enough for heredity and metabolism to strengthen. Once established, it can spread upward again when conditions improve. In that view, bombardment becomes a challenge that shapes where life first took hold, not an automatic end to the story. Some microbes can survive intense radiation by rapidly repairing DNA. Radiation can break DNA strands and scramble genetic instructions, which is usually catastrophic. Yet some microbes are astonishingly good at recovery, using coordinated repair systems to stitch broken DNA back together and correct damage. This resilience matters for early Earth because ultraviolet exposure was stronger before atmospheric shielding became robust. It also matters for any environment where radiation is a constant hazard, including high-altitude deserts and surface rocks exposed to cosmic rays. The deeper fascination is what repair represents. It is a way for life to keep order in the face of relentless disorder. Instead of needing perfect protection, an organism can survive by rebuilding quickly and accurately after damage occurs. That strategy echoes the broader origins story, where persistence is not about avoiding every problem. It is about coping, adapting, and continuing. In harsh settings, the winners may be those that can recover fastest, turning injury into a temporary detour rather than a dead end. Certain spores can endure years of dryness and wake when water returns. When conditions become difficult, some microbes form spores that are compact, protected, and metabolically quiet. In that dormant state, they can tolerate long, dry periods that would kill active cells, then revive when moisture returns. The protection comes from tough outer layers and internal chemistry that stabilizes vital components during the wait. For origins, this is a reminder that survival does not require constant activity. Early environments could swing between wet and dry, warm and cold, calm and chaotic. A lineage that can pause its biological clock gains time, and time is everything when evolution is still finding its footing. Dormancy also supports dispersal. Wind, dust, or flowing water can carry hardy spores to new locations, where different chemistry and new resources await. That helps explain how early life might spread across a young planet, not as a continuous wave, but as scattered seeds that awaken when the moment is right. Tardigrades survive extremes, but they still need life-friendly conditions to reproduce. These tiny animals can endure conditions that sound impossible, including deep freezing and intense dehydration, by entering a state where metabolism nearly stops. In that state, their bodies protect key structures and wait out the danger. The fascinating catch is that endurance is not the same as thriving. They still need suitable water, temperature, and food to grow and reproduce. This distinction is useful in origins thinking. Survival tricks can keep life present through hard times, but long-term evolution requires periods of genuine habitability, where organisms can multiply and explore new adaptations. Extremophile headlines can make it sound as if life can flourish anywhere, yet biology still depends on the basics. Energy, liquid water, and workable chemistry must be available often enough for populations to persist. Tardigrades remind us that resilience can buy time, but it cannot replace an environment that supports continued living cycles. Life's building blocks include phosphorus, which can be scarce in nature. Phosphorus is a quiet cornerstone of life. It helps form the backbone of DNA and RNA. It appears in ATP, and it plays structural roles in membranes. The problem is availability. Many phosphorus minerals are not very soluble, which can make usable phosphate hard to obtain in some environments. That creates a genuine origins challenge, because early chemistry would need a way to access phosphorus often enough to build larger systems. Researchers explore settings that could concentrate phosphate, such as certain volcanic regions, evaporating ponds, and mineral surfaces that collect and release it in cycles. They also look at alternative phosphorus compounds that might have been more reactive than ordinary phosphate. The intrigue here is that life's necessities are not automatically abundant. Origins are shaped not only by what is possible, but by what is accessible. A planet can have the right elements in bulk, yet still struggle to supply them in the right form, in the right place, at the right time. Some theories suggest lightning made usable phosphorus from common minerals. Lightning is not only a spark in the sky. It can be a furnace with temperatures high enough to melt and transform minerals in an instant. When lightning strikes certain rocks, it can create glassy structures called fulgurites. And in that violent chemistry, phosphorus can be converted into more reactive forms. Those reactive phosphorus compounds can dissolve more readily and participate in prebiotic reactions more easily than stable phosphate locked in minerals. The appeal of this idea is that it connects a rare but repeated event to a persistent problem. Storms happen, and over long time, their chemical effects can accumulate. Each strike could create a small local cache of usable phosphorus, which rainwater could then carry into ponds, streams, or wet sediments. It is a cinematic thought. A flash of electricity reshapes rock, then the next rainfall spreads the products into the landscape, feeding chemistry that needs phosphorus to build energy carriers and genetic backbones. Not every scientist is convinced this path was dominant, yet it is a vivid example of how planetary processes might unlock crucial ingredients. The phosphate problem is still one of Origin's research's big hurdles. This challenge persists because life needs phosphorus everywhere, yet early Earth chemistry may not have had easy access to enough of it in reactive forms. In many settings, phosphate binds tightly to minerals or becomes trapped in insoluble compounds, which limits how much can circulate in water. That forces Origin scenarios to be specific. They must point to environments that can gather phosphate, keep it available, and cycle it in ways that support building larger molecules. Some ideas focus on volcanic regions with unusual minerals. Others explore drying cycles that concentrate phosphate in small pools. Others consider alternative phosphorus chemistries that later gave way to modern phosphate, once biology evolved better ways to handle it. The fascination is that a single element can shape the entire plausibility landscape. Origins are not only about clever reactions. They are also about supply chains. The first living systems had to solve resource problems, and this one remains a key puzzle for scientists today. Life depends on nitrogen, and early Earth needed ways to fix it. Nitrogen gas filled the air, yet it was locked behind a stubborn triple bond that most chemistry cannot easily break. That creates a strange situation. The ingredient is everywhere, but it is not usable. Living systems need nitrogen in forms that can be built into amino acids, nucleic acids, and many other essentials. So a young biosphere would need a solution, either from geology, from lightning chemistry, or from early biological innovation. Some environments can produce reactive nitrogen compounds through heat and energetic events, but these supplies may be patchy and short-lived. That patchiness could shape where early ecosystems flourished, favoring places where usable nitrogen appeared often enough to sustain growth. The moment a lineage gained a dependable way to unlock nitrogen, it would gain a huge advantage. It would no longer wait for lucky deliveries. It could build continuously, spread into new habitats, and reshape the chemistry around it. Some microbes turn nitrogen gas into ammonia using an enzyme toolkit. To transform nitrogen gas into ammonia, a microbe must do something that feels like industrial chemistry, but inside a cell. The process requires careful control of electrons and protons, delivered in the right order, under the right conditions. The payoff is enormous, because ammonia can be fed directly into the building of proteins and genetic material. This ability is called nitrogen fixation, and it is one of the quiet foundations of ecosystems. Without it, life would run out of usable nitrogen even if the atmosphere stayed full of it. The most captivating part is that nitrogen fixation is costly. It demands energy, and the machinery is sensitive. That means a fixer must balance expense and benefit, and many organisms solve it through partnerships. Some bacteria live alongside plants, exchanging fixed nitrogen for sugars. In origin stories, this kind of chemistry suggests a major evolutionary leap, where life learned not just to adapt to its environment, but to unlock a global resource and make it biologically accessible. That enzyme uses metal clusters, like tiny industrial machines. At the heart of nitrogen fixation sits a protein complex that relies on tightly arranged metal atoms, often including iron and sometimes molybdenum ore. Vanadium. These metals are not decoration. They create an active center where electrons can be handed off with precision and where nitrogen can be coaxed into reacting. The idea is astonishing because it looks like engineered machinery, yet it is the product of evolution. The metal cluster helps weaken nitrogen's strong bond, step by step, while the surrounding protein controls timing and protects unstable intermediates. This is one reason Origins researchers pay attention to metals. If early Earth offered the right trace elements in certain niches, it could make advanced chemistry possible sooner. Metal availability becomes a kind of hidden map, guiding which environments could support powerful catalysts. When you picture the first complex enzymes, it helps to imagine them as refined versions of simpler mineral chemistry, now wrapped in proteins that hold atoms in power. Exactly the right arrangement. Sulfur chemistry may have been central in the first metabolisms. Sulfur comes in many chemical forms, and it is eager to trade electrons. That makes it useful as both an energy source and an energy sink, depending on the environment. In places where volcanic activity and hydrothermal fluids supply sulfur compounds, microbes today can thrive without sunlight by running their metabolisms on sulfur chemistry. That modern fact hints at an ancient possibility. Early life could have used sulfur-rich reactions because they are energetically favorable, and because sulfur compounds can be abundant near geologically active areas. Sulfur can also interact with metals in ways that create catalytic surfaces, which may help complex reaction networks get started. The appeal of sulfur in Origin scenarios is its versatility. It can support cycles, it can drive gradients, and it can link carbon chemistry to energy flow. If you want a world where chemistry can keep going without delicate ingredients, sulfur is a strong candidate. It offers robust pathways in environments that feel like early Earth. Trace metals can steer reactions, making some pathways more likely. A tiny amount of metal can change the fate of a whole chemical mixture. Metals like iron, nickel, cobalt, and zinc can bind to molecules and alter how electrons move, which can speed certain reactions and suppress others. In a messy prebiotic environment, that steering matters. Without guidance, chemistry can produce many dead ends. With a catalytic metal present, one pathway can become favored, creating a more repeatable outcome. This idea connects Origins to local geology. Two ponds with similar water can behave very differently if one is fed by metal-rich rock and the other is not. Trace metals also help explain why modern enzymes often keep metals at their core. Biology may have learned early that metals are reliable helpers, then evolved proteins that hold those metals in precise shapes. In the story of life's beginnings, metals can act like quiet directors. They do not create ingredients from nothing. They nudge the chemistry that already exists toward patterns that can persist, spread, and eventually evolve. The earliest life was probably heat-loving, based on many family trees. When scientists compare genetic relationships across many microbes, some analyses place heat-tolerant organisms near the base of the tree. That suggests early life may have thrived in warm environments, perhaps near hot springs or hydrothermal systems. Heat can speed reactions, which helps chemistry build complexity, and warm water can keep certain minerals and gases more available. Yet heat is also challenging, because it can break fragile molecules. So if early life did live hot, it likely had ways to stabilize its components, or it relied on robust chemistry that could withstand stress. A warm origin setting also fits with a planet that was more geologically active and possibly more volcanically restless than today. Imagine a world with abundant hot water circulation through rocks, creating steady gradients and constant chemical turnover. In that world, being heat-loving is not a quirky specialty. It is the default. If early ecosystems began there, later evolution could expand outward into cooler and more varied habitats as chemistry and heredity became more reliable. Yet some evidence points to cooler starts, so the debate continues. Not all clues point toward a hot beginning. Some chemical signatures and experimental pathways work better at moderate temperatures, where delicate molecules last longer and unwanted side reactions slow down. Cooler settings can also provide natural concentration methods, such as freeze-thaw cycles that push molecules into tight liquid channels. In addition, some models suggest that certain early polymers and membranes are more stable in milder conditions than in near boiling water. This tension creates a productive debate. If life began hot, it gains fast chemistry and energetic geology. If it began cooler, it gains stability and longer molecular lifetimes. The answer might even involve a sequence. Chemistry could begin in one setting, then become more lifelike in another. The debate matters because it guides where we look for plausible environments, both on early Earth and on other worlds. If a warm start is required, we focus on hydrothermal activity. If a cool start is possible, the range of candidate habitats expands dramatically, including icy worlds with hidden oceans. Even defining life is tricky when chemistry starts to self-organize. At the start, the boundary between living and non-living can look more like a gradient than a line. A crystal can grow, yet it is not alive. A fire spreads, yet it has no heredity. A chemical network might sustain itself for a while, concentrate its own ingredients, and respond to changes, yet still fall short of what we call life. Origins research forces you to ask what you really mean by living. Is it metabolism, a steady use of energy to maintain structure? Is it compartments, a protected inside with its own chemistry? Is it information, stored patterns that can be copied? Or is it evolution, where improvements accumulate over generations? Different definitions highlight different milestones, and each milestone suggests different environments that could support it. The fascination is that life may not have arrived in one leap. It may have arrived as a sequence of achievements, each one making the next easier. In that view, the earliest stage might not look like a tiny cell. It might look like chemistry that has started to behave with purpose, even though no mind is present. Some researchers use Darwinian evolution as the key threshold. One useful way to draw the line is to ask a simple question. Can a system evolve through selection, where variations compete and the winners become more common? If the answer is yes, then the system can improve over time without needing a designer. That is the essence of Darwinian evolution, and it is why some scientists treat it as the moment life truly begins. Before that threshold, chemistry can be interesting, even complex, yet it cannot build a cumulative history. After that threshold, small advantages can stack, and the system can climb toward greater capability. The challenge is that Darwinian evolution requires both variation and some form of inheritance. Something must be copied well enough that improvements are not lost immediately. That could be genetic sequences, or it could be compositional patterns in protocells that are passed on when containers divide. The moment such inheritance becomes reliable enough, the story accelerates. Evolution becomes an engine, turning planetary chemistry into a lineage that can explore possibilities far beyond its humble beginnings. The first true cells needed reliable inheritance, not just chemistry. At some point, a living system had to do more than persist. It had to pass its success forward. A protocell that grows and divides is impressive, yet if each division scrambles what made it work, progress cannot accumulate. Reliable inheritance means that useful instructions, or useful internal patterns, survive the handoff from one generation to the next. That pressure would favor anything that stabilizes information, such as stronger polymers, better copying helpers, or compartments that keep key ingredients together. It would also favor routines that prevent the cell from drifting into randomness, like controlling what enters and leaves and keeping reactions in a repeatable order. Once inheritance becomes steady, evolution gains memory. Improvements stop being one-time accidents. They become part of a lineage. That is when biology begins to feel inevitable, because success can finally be stored, repeated, and refined. Error rates in early copying had to drop below a critical limit. Imagine trying to copy a long message while making mistakes on every line. You might still get something readable at first, but as the message grows, errors overwhelm meaning. Early genetic copying faced a similar constraint. If copying was too inaccurate, information could not be maintained, especially as sequences became longer and more useful. This creates a threshold problem. Below a certain error level, a system can preserve and build complexity. Above it, the system collapses into noisy variation and cannot improve. The fascinating implication is that early evolution may have been trapped in a narrow corridor. It needed copying that was good enough to hold on to advantages, yet simple enough to arise without advanced enzymes. Researchers explore helpers like metal ions, protective environments, and cooperative networks that make copying steadier. Once accuracy improved, longer sequences could survive. Longer sequences could encode better tools. Better tools could improve accuracy further. That feedback could accelerate the transition from chemistry into true heredity. Natural selection can improve replication long before modern biology appears. You do not need a full cell to get selection. You only need variation, competition, and a way for successful patterns to persist. In a prebiotic setting, that could mean short polymers that form in slightly different sequences, where some sequences happen to last longer or help assemble copies more effectively. It could also mean simple reaction networks where certain pathways reinforce their own ingredients, allowing them to outlast rival pathways when resources are limited. The key is that small advantages can compound. A molecule that makes slightly more of itself will become more common. Once it is common, it is more likely to encounter partners, build larger structures, or occupy more favorable microenvironments. Over time, replication can be refined by this quiet sorting process, step by step. This is what makes origins feel less like a single miracle. The earliest winners might have been crude and inefficient, yet selection can gradually polish crude beginnings into something that behaves more and more like life. Compartments help selection by keeping winners and losers separated. In open water, a helpful molecule can benefit its neighbors just as much as itself. That makes it hard for selection to reward the right chemistry. A compartment changes the rules. When molecules are enclosed together, the outcomes of their reactions stay local. If one protocell contains a set of reactions that helps it grow, it gains more membrane material, divides more often, and leaves more descendants. A neighboring protocell with less effective chemistry may shrink, leak, or fail to reproduce. This creates a clean stage for competition, where success is linked to a physical container. Compartments also allow experimentation. Each tiny bubble can run a slightly different chemical program, and the environment can sort them by performance. Some will be dead ends. Others will persist long enough to be improved. Once you have that, evolution has a handle. It can favor systems that keep useful combinations together, which is a major step toward the organized complexity we associate with living cells. Early life likely fed on geochemical energy from Earth's interior. Before food webs and oxygen, a young biosphere would still need a dependable energy supply. Earth's interior provides one through chemical disequilibria, where reduced compounds from deep rock meet more oxidized conditions near the surface. When those chemical differences interact, energy is released, and that energy can be captured by metabolism. Think of it as a planet that is still cooling and reacting, offering fuel lines that do not depend on sunlight. Such energy sources can be steady over long times, especially in regions with active circulation of water through crust. This matters for origins because steady energy supports persistence. It allows reaction networks to keep running, protocells to keep cycling, and small improvements to matter. It also makes the search for life beyond Earth more interesting. If a world has water and rock interactions, it may have energy even under ice or in darkness. Life's earliest diet may have been the planet itself, served through chemistry. Today, deep subsurface microbes still live kilometers beneath ground. Far below your feet, there are organisms living in cracks, pores, and thin films of water, in places without sunlight and with painfully limited nutrients. Some survive on slow chemical reactions between water and rock, and they can persist at rates that make surface life seem frantic. Their existence expands what we consider a habitable world. It also offers a living analogy for early Earth, when surface conditions could be unstable and protective niches would be valuable. In the subsurface, temperatures rise, pressures increase, and the environment is buffered against rapid change. That stability can support long-term survival, even when growth is slow. Deep microbes also reshape geology in subtle ways, altering minerals and influencing the chemistry of groundwater. For origins thinking, this matters because it suggests life can occupy hidden spaces for immense stretches of time, quietly accumulating adaptations. If early life ever retreated underground during harsh surface periods, it could have endured, evolved, and later re-emerged to repopulate more welcoming, quote, habitats. Some ecosystems rely on hydrogen made when rocks react with water. In certain environments, hydrogen is produced naturally when water interacts with specific minerals in the crust. That hydrogen can become the energy backbone of an ecosystem, feeding microbes that combine it with carbon compounds and turn that energy into growth. What is striking is the independence of this food chain. It does not begin with sunlight. It begins with geology. In vent regions within oceanic crust, and in some continental settings, hydrogen can seep into surrounding waters, and microbial communities can gather bio. Where the supply is strongest, these organisms are often masters of scarcity, extracting energy from reactions that seem modest yet are dependable. This kind of ecosystem is exciting for origins and for astrobiology because it shows a pathway that can operate in darkness, under ice, or deep, underground. If a planet or moon offers water rock reactions that generate hydrogen, it may also offer a foothold for life. Hydrogen becomes a whisper of possibility, carried by stone and water. Serpentinization can produce hydrogen and simple organics naturally. When certain mantle-derived rocks react with water, their minerals change structure and chemistry. This process, called serpentinization, releases hydrogen as a byproduct, and it can also support the formation of small organic molecules under the ID. Right conditions. The fascination is that it links planetary geology to lifelike chemistry without requiring biology to start the chain. The reactions can also raise the alkalinity of surrounding fluids, creating distinctive chemical environments that persist as long as water continues to circulate through rock. Over time, serpentinization can build a steady chemical landscape with energy-rich molecules and mineral surfaces that can catalyze further reactions. This makes it a favorite mechanism in origins discussions because it is not a rare event. It is a repeating geochemical engine. It also appears to be plausible on other worlds with water and rocky interiors. In that sense, serpentinization is not just an Earth story. It is a planetary recipe that could run in many places, quietly producing fuel and starting materials in the dark. Alkaline vents create conditions that resemble parts of cell chemistry. In some vent systems, fluids are strongly alkaline, and they meet relatively more acidic seawater across mineral structures. This sets up differences in pH and chemistry that can be maintained for long periods. What makes this so tantalizing is how closely it echoes a core strategy used by cells, which is harvesting energy from gradients across boundaries. In an alkaline vent setting, the boundary is mineral rather than biological, yet it can still separate environments and guide ion movement. The mineral pores can also concentrate molecules and provide catalytic surfaces, which helps reactions proceed in a more organized way. This combination, steady gradients plus structured microspaces, gives Origins researchers a coherent stage for early metabolism-like processes. It offers a way to get direction, persistence, and concentration without needing advanced enzymes or complex membranes at the start. The vent becomes a natural reactor with built-in architecture, where chemistry can be driven repeatedly toward greater complexity. The boundary between rock and water may have life's first home. Interfaces are where chemistry gets interesting. In open water, molecules drift apart. On a rock surface, molecules can stick, concentrate, and meet again and again. The rock can also create microenvironments, small pockets with different acidity, different ion balance, and different access to catalysts. Water brings mobility and mixing, while rock brings structure and stability. Together they create a setting where reactions can be both repeated and shaped. Many origin scenarios, even when they disagree on details, return to this meeting point. It is a place where energy gradients can exist, where minerals can assist transformations, and where compartments can begin as pores or films before becoming shear, true membranes. It is also a place that exists in countless forms, from seafloor cracks to shoreline sediments to hot spring terraces. If life began at an interface, it began in a place where the planet itself could guide chemistry, not by intention, but by providing surfaces, cycles, persistent boundaries that make complex reactions more likely to take hold. Life's earliest fossils are subtle, and scientists argue about some claims. The oldest traces of biology are rarely neat bones or shells. They are whispers in stone, faint textures, tiny shapes, or chemical patterns that might have many explanations. Heat and pressure can squeeze and distort ancient rocks, and flowing fluids can rewrite them from the inside. That makes early evidence feel like a crime scene that has been cleaned up by time. Researchers look for multiple clues that agree, such as microscopic structures that sit in the right setting, paired with chemistry that fits biological activity. Even then, debate is healthy, because geology can be an excellent imitator. The most exciting part is that disagreement does not mean failure. It means the questions are sharp. Each contested claim pushes new methods, better imaging, and stricter standards. When a signal survives that scrutiny, it becomes more than an interesting idea. It becomes a rare, hard-won window into Earth's deepest biological past. Carbon isotopes can hint at biology, but geology can mimic them. Carbon comes in slightly different forms, and life often favors the lighter version because it is easier to process in many reactions. That preference can leave a measurable fingerprint in ancient rocks. It is like finding a pattern that suggests a familiar hand was at work. The catch is that non-living processes can sometimes create similar patterns, depending on temperature, pressure, and the history of fluids moving through rock. That is why carbon isotope evidence is powerful, yet never enough on its own. Scientists try to link isotope signals to context. They ask what kind of rock it is, how it formed, and whether it has been altered since its birth. They compare the signal with other chemical indicators and with the microscopic fabric of the sample. When multiple lines of evidence align, isotopes become thrilling. They can suggest life's presence even when no clear fossil shape exists. They can also reveal how early metabolisms may have processed carbon in a world unlike today. Microfossils can be faked by minerals, so confirmation needs many tests. A tiny filament in rock can look like a cell chain, yet minerals can grow into similar shapes through purely chemical processes. Crystal growth can produce tubes, spheres, and branching forms that seem biological at first glance. That is why a single photograph, no matter how compelling, cannot settle the question. Researchers treat candidate microfossils like fragile evidence that must survive cross-examination. They examine the surrounding rock layers to see if the environment makes biological sense. They analyze chemistry to check for carbon where it should be, and for minerals that form in ways consistent with life. They use multiple imaging techniques to see whether the structure is hollow, layered, or composed of mineral grains that signal a non-living origin. The goal is not to dismiss exciting finds. It is to protect the story from wishful thinking. When a candidate passes many independent tests, the result is electrifying. It means something alive likely once shaped matter on a young Earth. Some of the oldest candidate microfossils are fiercely debated today. A few famous samples have sparked years of discussion because they sit close to the edge of what rocks can reliably preserve. The structures can be extremely small, and the rocks can be altered by long geological history. That creates space for two honest interpretations. One side sees features that resemble microbial life, placed in a plausible ancient environment. The other side sees structures that could arise from mineral growth, deformation, or chemical alteration. What makes these debates so captivating is that they are not just arguments about one rock. They are arguments about standards. How much evidence is enough to claim life at the extreme edge of time? Each new instrument can shift the conversation because better resolution or new chemical mapping can reveal details that were invisible before. This is how science earns trust. It does not rush. It repeats, challenges, refines, and sometimes reverses. In the end, even disputed candidates are valuable. They sharpen the tools we will need to recognize life on other worlds, where the evidence may be even harder to read. Genetic studies suggest life arose quickly after Earth became habitable. When scientists compare genes across the tree of life, they can estimate how far back major lineages diverged. These estimates depend on assumptions, and they come with uncertainties, yet they often point to a surprisingly early origin. The logic is simple. If all modern life shares deep common features, and those features are already complex, then their roots likely extend far back into early Earth history. That hint is fascinating because it suggests life may not require a perfect, once-in-our-universe stroke of luck. It may emerge when conditions allow chemistry to run long enough, with enough energy and the right ingredients. Still, genetic timing is tricky. Mutation rates can change over time. Gene swapping can blur early branches. Extinctions can erase entire chapters. So the story is not a precise date. It is a pattern that keeps returning. Once Earth cooled and stable water environments existed, biology may have arrived on a timescale that feels fast for geology. That possibility adds a hopeful tone to Origins research because it implies that life might be a natural outcome on many wet, active planets. The earliest oceans formed soon after Earth cooled from its molten youth. After Earth's violent beginning, the surface eventually cooled enough for water to condense and collect. That transition matters because water is not only a solvent, it is a stage where molecules can move, meet, and react. An early ocean also becomes a planetary mixer, carrying heat and chemistry across huge distances. It can dissolve gases, interact with rock, and create countless microenvironments from deep seafloor to shallow shoreline. The most striking idea is how quickly a planet can shift from glowing chaos to liquid stability. Once water is present, weathering begins, minerals leach into the sea, and cycles start to form. Those cycles can create chemical gradients, concentration zones, and pockets where organics can accumulate. An ocean is not a single environment. It is a collection of environments stitched together by currents and tides. For origins, that variety is precious. It means early Earth did not need to find one perfect location. It had millions of possibilities, spread across a global ocean that kept reshaping the chemistry of its own shores. The moon-forming impact may have reset Earth's surface conditions. One leading idea is that a colossal collision early in Earth's history helped create the moon. If that happened, it would have been an event that profoundly altered the surface, the atmosphere, and the planet's thermal state. For origins research, a reset matters because it frames the timeline. If conditions were extreme for a while, early chemistry may have had to begin again, or find refuge in protected niches. Yet a reset is not only destruction. It can also create new starting conditions. It can influence how quickly the planet cools, how the crust forms, and how oceans and atmospheres evolve afterward. It may also help explain why Earth has strong tides and a stable axial tilt, both of which shape long-term climate behavior. The intriguing part is how a single event can cast a long shadow. Even if life began later, the environment it inherited may have been sculpted by that ancient collision. In that sense, the story of life's beginnings may include a planetary drama before biology ever appeared, where the moon's birth helped set the stage for everything that followed. Tides were likely stronger, mixing shorelines and chemical ingredients. A closer moon would have pulled more strongly on Earth's oceans, and stronger tides mean more vigorous movement along coastlines. That movement matters because shorelines are natural chemistry engines. They collect material from land, concentrate it in shallow water, and constantly stir it with incoming and outgoing flows. Strong tides can repeatedly wet and expose sediments, transport minerals, and mix different water chemistries that would otherwise stay separated. They can also create small pools that fill and drain, which changes concentration and can drive reactions that need fluctuating conditions. This is a very physical kind of assistance. No rare spark is required, only the daily rhythm of water moving back and forth. A tide is like a metronome for early chemistry, providing repetition and mixing on a schedule the planet can keep for millions of years. When you picture origins, it is easy to focus on exotic vents or lightning. Tides remind you that ordinary, persistent motion can be just as important. The shoreline can become a workshop, and the moon can be one of its quiet power sources. Wet and dry cycles can drive polymer formation better than constant water. Many of the molecules life depends on are polymers, long chains built from smaller units. In continuously wet conditions, building long chains can be frustrating because water encourages bonds to break and makes it harder to link units together. A wet and dry cycle changes that. During drying, molecules become concentrated and reactions that join units can become more favorable. During rewetting, products can disperse, fold, and interact in new ways, and fresh ingredients can be brought in for the next round. This creates a natural rhythm of assembly and testing. It is also a form of selection. Chains that survive drying and rehydration become more common in that environment. chains that fall apart are filtered out. Over many cycles, the chemistry that persists can shift toward greater robustness and complexity. What makes this so compelling is how ordinary it is. You do not need a rare event. You need a place where water comes and goes, like a shoreline shelf, a seasonal pool, or a hot spring basin. Repetition becomes the engine, and time does the rest. Modern cells still use chemistry that looks like patched-together ancient parts. Inside any cell, you can find layers of history stacked like renovations in an old house. Some reactions look sleek and specialized, while others feel like versatile workarounds that kept being reused. Many pathways share small molecular helpers that appear again and again, as if evolution preferred familiar tools over reinventing everything. When a new problem appeared, life often solved it by borrowing a piece that already worked and wiring it into a new context. That kind of reuse leaves fingerprints. The same cofactor can appear in unrelated processes. The same chemical trick can show up in different corners of metabolism. This matters for origins because it suggests early biology was not designed as a clean system. It grew by accumulation. It kept what functioned, replaced what failed, and layered improvements on top of earlier solutions until the result looked seamless. The citric acid cycle has branches that may trace back to early metabolism. This famous cycle is often taught as a loop that helps cells harvest energy, yet it is also a hub for making important building blocks. Many organisms use pieces of the cycle without running the whole loop in the textbook direction. Some pathways run portions in reverse to build carbon-rich molecules from simpler inputs, which is especially useful for organisms that make their own gas, biomass from gases. That flexibility is why some researchers suspect parts of the cycle reflect very old chemistry. A small set of reactions that reliably builds useful intermediates would be valuable long before complex cells existed. Over time, those reactions could be connected and refined into a more complete network. What began as a few reliable steps could become a central roundabout, feeding amino acids, fats, and other essentials. It is a reminder that metabolism may have started as fragments, then gradually assembled into cycles that look elegantly complete. Many key enzymes rely on metals, echoing mineral chemistry outside cells. Some of life's most important reactions are guided by atoms that life did not invent. Metals can hold charges, shift electrons, and stabilize awkward intermediate states that would otherwise fall apart. Inside proteins, metals act like precision tools, enabling reactions that pure organic chemistry struggles to do quickly. Magnesium often helps manage phosphate chemistry in processes tied to genetic material and energy transfer. Zinc can stabilize structures and assist reactions that reshape molecules. Copper and iron can help move electrons in controlled steps. The most intriguing connection is geological. Early Earth offered a rich menu of metal ions and mineral surfaces, and those could have nudged prebiotic chemistry toward useful pathways. Later, evolving proteins could capture those same metals and hold them in consistent positions, turning a messy environmental helper into a dependable internal machine. In that sense, enzymes may preserve a partnership between biology and the planet's mineral heritage. The core of the ribosome is ancient and mostly made of RNA. It is one thing to say life evolved. It is another to find a molecular structure so universal that it feels like a relic that never left. This central machine links amino acids into proteins, and it sits at the heart of every living cell's ability to build itself. Its deepest core is remarkably conserved across life, which suggests that once this system emerged, it became too valuable to replace. That conservatism matters for origins because it hints at a time when biological innovation had to be compatible with an already established translation system. Early evolution may have revolved around improving accuracy, speed, and control, while keeping the fundamental mechanism intact. By studying which regions are most conserved, scientists can infer what was essential early on and what was added later as refinements. The ribosome becomes a kind of molecular fossil record, not in rock, but in working geometry that has endured across deep time. Life's universals suggest early evolution favored what worked, not what was perfect. Biology is full of choices that seem odd if you imagine a clean engineering project. Many solutions are good enough rather than ideal, and they persist because changing them would break too much at once. This is why shared features across life are so revealing. When nearly everything uses the same core strategies for storing information, building proteins, and managing energy, it implies those strategies took hold early and then became locked in. Early evolution likely happened under pressure to survive, not to optimize elegance. If a pathway allowed faster growth or better stability in a particular environment, it could become entrenched, even if another pathway might have been theoretically superior. Once many other systems depended on it, switching became dangerous. That kind of lock-in turns early accidents into long-term foundations. It also makes origins feel more natural. The first living systems did not need to be flawless. They needed to be viable, repeatable, and capable of improvement. Some origin experiments create self-replicating chemical systems, but not full life. In laboratories, researchers can build chemical setups where certain molecules help produce more of themselves, or where networks of reactions reinforce their own keen. Continuation. These systems can show growth-like behavior, competition-like dynamics, and even a limited kind of heredity. Yet they also reveal how many abilities modern life quietly bundles together. True life must do more than replicate. It must manage energy, maintain itself against decay, and respond to changing conditions while keeping information meaningful. Lab systems often excel at one aspect while lacking others. That gap is not discouraging. It is illuminating. It tells us which steps might be easier and which are the hard bottlenecks. It also encourages creativity. Researchers try different environments, different catalysts, and different cycles of heating, drying, and mixing to see what new behaviors emerge. Each partial success is like finding a stepping stone in fog. It does not cross the river alone, but it shows the river has places to stand. We still do not know the exact path from molecules to organisms. There are many plausible routes, and that is part of what makes the question so captivating. Different environments can produce different ingredients. Different ingredients can support different kinds of networks. Some scenarios begin with information, others begin with metabolism, and others begin with compartments and let the rest follow. The difficulty is that early Earth left only scattered traces, and chemistry offers many ways to reach similar outcomes. Two very different beginnings could, in principle, converge on cells that look familiar today. That uncertainty is not a weakness of the field. It is a sign that we are trying to reconstruct a deep past with limited clues, while also respecting how creative chemistry can be. Each new experiment adds one more constraint. Each new geological discovery adds one more boundary condition. Over time, the space of possibilities shrinks. The path is still unknown, yet the map is getting sharper, and the search itself keeps revealing surprising connections between rocks, water, and living processes. That mystery makes life's existence feel even more improbable and precious. When you realize how many steps must align, it is hard not to feel a quiet awe. Chemistry must produce the right kinds of molecules. Those molecules must persist long enough to interact. Energy must be available in a form that can be harnessed, not just released. Information must become stable enough to accumulate improvements. All of this must happen in a real environment that changes, disrupts, and sometimes destroys. And yet, somewhere in that complexity, the first lineage found a way to continue. The mystery does not only make life feel unlikely, it also makes it feel meaningful in a very physical way. You are not separate from the story. Your body is what happens when that ancient continuity never broke. Every cell carries the result of countless survival decisions, layered over unfathomable time. Origins research can feel abstract, yet it points back to something intimate. Existence is a chain of successful handoffs, from the first workable chemistry to the living world you inhabit right now. Every new origin idea must explain both chemistry and evolution together. It is not enough for a scenario to produce interesting molecules. It must also explain how those molecules become part of a system that can improve. A convincing idea needs a source of ingredients, a source of energy, and a setting that concentrates and protects key reactions. Then it needs a mechanism for variation, persistence, and selection. If the chemistry is rich but cannot be inherited, progress cannot accumulate. If heredity is present but the chemistry cannot supply materials or energy, the system stalls. This is why the best origin models feel like complete stories rather than isolated tricks. They connect environment to mechanism and mechanism to long-term change. They also face hard questions about plausibility. Could the necessary conditions exist often enough? Could the system survive disturbances? Could it spread beyond its birth site? The demand for both chemistry and evolution is a useful filter. It keeps the field grounded while still leaving room for surprising solutions that blend geology, physics, and biology into one continuous narrative. The search for life's origins also guides how we hunt for life elsewhere. Origins research teaches us what signs might matter most on other worlds. Instead of looking only for familiar creatures, scientists look for environments that could sustain chemistry over time. They look for liquid water, energy gradients, and cycles that concentrate materials. They consider whether a planet or moon has active geology, because rock and water interactions can supply chemical fuel even without sunlight. They also learn caution from Earth's own subtle record. Early life did not announce itself with obvious skeletons. It left indirect fingerprints that require context to interpret. That lesson shapes missions and instruments, from sampling rocks to analyzing atmospheric gases. It also expands imagination. If life can begin in dark settings powered by chemistry, then icy moons with hidden oceans become compelling targets. If life requires certain ingredients to be available in usable forms, then mineralogy matters as much as temperature. In that way, studying our own beginnings becomes a practical guidebook for exploration, turning an ancient mystery into a map for where to search next. As we come to the end of our journey tonight, it can be comforting to notice how wide the story has been. We began with raw chemistry, with rocks, water, energy, and time. We wandered through oceans that changed color, skies that looked unfamiliar, and shorelines that rose and fell with strong tides. We followed molecules as they learned to gather, to persist, and to pass their patterns forward. Step by step, the world discovered how to keep a memory of what worked. Along the way, we saw how life borrowed from its surroundings. It used minerals, gradients, metals, and cycles that the planet already offered. Nothing appeared fully formed. Everything built slowly, layering success upon success, until chemistry became biology, and biology became a living history that still flows through you now. You might imagine those early environments settling into the dark, the slow movement of water through rock, the steady rhythm of tides, the warmth of vents, and the long patience of time itself. Let that vastness soften your thoughts. You do not need to hold every detail. The story can drift, like sediment settling after a long current. If you find yourself still awake and curious, there is another video waiting for you on the screen, ready to carry you into a new, quiet exploration. And if these nighttime journeys have brought you comfort or wonder, you are always welcome to support the channel by liking, subscribing, or sharing a thought. It helps these stories reach others who may need them too. For now, allow your breathing to slow. Let your body rest where it is. You have traced the path from stardust to cells, from stone to story. There is nothing left to follow tonight. Sleep well, and good night.